Good morning. Today in this video, we are going to recapitulate what we have started last time on osmosis and then go over the rest of the slides on osmosis. As we said before, osmosis is a passive process. Why is it a passive process? Because it does not require energy. And why doesn't it require energy? Because as usual, water molecules will pass from where they are present in a higher concentration to where they are present in a lower concentration. So it looks like simple diffusion. But why is it called osmosis in particular and not just simple diffusion? For two reasons. Because osmosis is a special type of diffusion that concerns water only. So we never mention the word osmosis unless we are talking about the passage of water molecules from where they are present in a higher concentration to where they are present in a lower concentration. And as we said before, it's not enough to say higher concentration, because if you just say higher concentration of a solution, for example, then you're talking about the concentration of the particles in that solution, of the solid particles in that solution, and not of water. So we should say from higher water concentration to lower water concentration. This is, or this applies if we're talking about water. But if you talk about a solute, you just say from higher concentration to lower concentration. What does it mean? Concentration of a solute. We have different types or different ways of defining concentration of a solute. For example, we can define it as mass per unit volume. So we can say, for example, that the concentration of sodium chloride in a solution in water is 10 grams per liters. Or if the concentration is higher, we can mention it in kilograms per liter. Or if the concentration is lower, we can mention it in milligrams per liter. So this is one way of expressing or defining concentration. But there is another type or another way of defining concentration, which is to mention the number of particles per unit volume. And this is what we usually use whenever we explain the concept of osmosis. So if you are in the lab, you will be using gram per liter or milligram per liter. But if you are explaining the concept to your students, for example, just like I'm doing now, you express it in the form of particles per unit volume. This is the first special feature of osmosis, that osmosis concerns only water. But there is another feature of osmosis other than the fact that it concerns water, is that we never see osmosis, or we can never understand osmosis perfectly unless there is a selectively permeable membrane between the two containers, the one or the two parts of the container actually, the side that contains a higher water concentration and the side that contains a lower water concentration. For example, here in that diagram, you can see that solution A and solution B are in the same container, but that the container is divided into two halves and that it is divided by a partially permeable membrane. That partially permeable membrane has pores, but these pores have a size that allows only water molecules to pass, which are the blue small ones, but prevents sugar molecules from passing, the ones that are bigger in size and that are red in color. That's why we say that it is a partially permeable membrane. You can also find the word semi-permeable or semi-permeable membrane, meaning that it allows the passage of certain molecules and or particles and prevents the passage of others. So it's not fully permeable. But if you use the word selectively permeable, so it includes the meaning of partial or semi-permeability, but adds to it the fact that the membrane will select. Here in partially permeable and semi-permeable membranes, the membrane just selects depending on the size of the pores. So it's a purely mechanical process. But when you say selective, so you can use partially and semi-permeable here, for example, when you do this experiment in the lab. But usually when you say selectively permeable, there is the concept of not only mechanical selection, but the concept of smart selection, meaning that you are talking, for example, about the cell membrane of the cell membrane that surrounds the cell, because there is some kind of intelligence, not only in the passage of water molecules from one side to the other, but in the passage of ions, for example. So the membrane will select what type of ions will pass and what types of ions will not pass according to the presence of channels. So when it comes to water and osmosis, you can use partially permeable or semi-permeable. But when you use selectively permeable, this is more when you talk about particles that are carried by carriers and channels where the decision is a smart decision. Coming back to solutions, we are talking about concentrations of solutions. So what is a solution actually? A solution consists of a solvent and a solute. The solvent is a liquid substance where the solid particles will dissolve. For example, if you have water and you add a teaspoonful of sugar, for example, or salt to that water. So in such a case, the solid particles of salt or sugar will be called the solute, whereas the liquid water will be called the solvent. But when we say that we have a solution, a true solution, then this means that salt or sugar particles will disperse themselves in between the molecules of water so that afterwards they would no more be soluble. So if you have a true solution, you will have a clear solution, meaning that a while after, you will see in the container a liquid that looks as if it was pure water, but actually it is a solution of salt or a solution of sugar, unless you add more and more salt and water, where the surplus of salt, and water will, the before reaching the surplus, there will be complete dissolution, but the surplus will settle at the bottom, although sugar or salt particles are capable of dissolving. But because you gave the water more than it can accommodate, and therefore in such a case, if you reach that state, we call this solution a supersaturated solution, meaning that you kept adding sugar or salt until the spaces in between the water molecules can no more accommodate the solid particles. And then we come to hyper and hypo. When we say hyper, we mean higher or more. And when we say hypo, we mean lower or less. So you cannot use, as we said in the previous video, you cannot use hyper and hypo without comparing two solutions. So what is a hypertonic solution? A hypertonic solution is a solution that contains more solute particles than the other solution. So for example, if this is solution A and that is solution B, then definitely solution B is hypertonic in relation to solution A. Meanwhile, solution A is hypotonic to solution B. So when we talk about concentration, we're actually talking about solute concentrations. And when we use the words or the terms hyper and hypo, we are comparing two solutions together. And of course, if the concentration of solute or solid particles on both sides is equal, then it would be a hypotonic solution. One of the important concepts that we have to understand, and because we're going to use frequently, is the osmotic potential. And the term osmotic potential, you can use another term that is equivalent, and that is called the solute potential. So actually, when we talk about osmotic potential, we are talking about the ability of solute particles to attract, to cause osmosis, or that's why the two nomenclatures or the two scientific terms apply. So it is solute potential because it is related to the ability of the solid particles. The ability of the solid particles to do what? To be a trigger for water to move by osmosis from one side of the container or from one solution to another. And we will be explaining this in detail. So osmotic potential or solute potential has a symbol which is psi s. This Greek letter, which looks like a W in English, is called psi, and psi s means that it is related to the solute. And actually, osmotic potential or solute potential is a component of a bigger quantity, which is called water potential, meaning that water potential includes solute potential plus something else. And this is what we mean by the fact that psi s or solute potential or osmotic potential is part of a bigger quantity called water potential. So water potential is more inclusive than solute potential. What's the meaning of solute particles triggering water to pass by osmosis? If we move back to this, we will see that the more you have actually the solid particles here, sorry, sorry, sorry for the delay. If we look at that slide, let me first check that we are still recording. Okay, so if you look at that slide, the presence of solid red particles here prevented this side of the container, which is side B, prevented this side of the container, which is side B, from having or accommodating a large amount of water. And the more you have of the solid particles on side B, the higher the solute concentration, the fewer water molecules available on side B because of the smaller space, and therefore this will be a trigger for those molecules to pass from side A to side B, because on side A, the number of water molecules is very high, whereas on side B, it is very low. So the more you have of the solute on one side, the more the ability of the water molecules to move. And in which direction will the water molecules move? They will move from side A, where we have more water molecules, to side B, where we have less water molecules. So it is a measure of the potential, or that's why we call it a solute potential or osmotic potential, because it is the ability or the capacity of the water molecules to pass from a hypotonic solution to a hypertonic solution across a semi-permeable membrane. And of course, it is related to solute concentration. So this osmotic potential, which is the ability of the water molecules to pass from the hypotonic side to the hypertonic side, will increase as you increase the solute concentration on the hypertonic side. It will increase, so it is directly proportional as you increase the solute concentration on the hypertonic side. So the more the solute concentration or the solute potential on the hypertonic side, the more the osmotic potential and the passage of water from hypotonic to hypertonic. If you have pure water, of course, pure water does not contain any solid particles. Therefore, its solute potential is zero because it has no solutes. Pure water has no solutes. Therefore, if it has no solutes, it sounds logical that it will have zero potential, zero solute potential, and therefore it will have a zero osmotic potential. This fact that osmotic potential is of pure water is zero is very important to solve the problems that you might encounter on osmosis. Then, if pure water has zero osmotic potential, then any solution will have a negative osmotic potential because the osmotic potential will be definitely less than zero because the presence of the solutes makes the water molecules less free to move due to the attraction forces. So if we come back here, the more you have of the solute on side B, the more you have of the solute on side B, the more the attraction forces, it expresses on what? On the water molecules. But which water molecules? On the water molecules on side B, on side B. So the more you increase the solute on side, this is very important but a bit difficult. So we have to focus and we have to be very careful when writing. So the more you increase the solute particles on side B, the more the attraction forces. Why? Because if this is sodium chloride, as you know, and as we said in biochemistry, since the water molecules are polar, having a partially positive and a partially negative side, then there will be attraction forces between the sodium chloride and water. The positive sodium will be attracted to the negative oxygen or to the partially negative oxygen, whereas the negative chloride will be attracted to the partially positive hydrogen side. How does this imply on the osmotic potential of or the ability of this particular solution B? Of course, this particular solution B, the water in it, will be more crippled and will not be allowed to move. And that's why it is less than in pure water. If we consider that pure water potential is zero, then the osmotic potential of a solution that has a higher solute concentration will be considered negative. But in such a case, it will increase the ability of the other solution A to move. Why? Because the more crippled these molecules are, the more this solution A or the water particles of that solution A will be capable of moving to the other side. So in order to wrap up, if we call this solution A and that solution B, solution A has an osmotic potential or a solute potential of 0. Why? Because it has no solid particles at all. Whereas solution B has a solute potential that is less than 0. That is a negative value. We can say, for example, -5. Okay? So this is the solute potential of solution A, and that is the solute potential of solution. So the more you increase the solid particles in solution B, the more water molecules that will be crippled, but the attraction forces between the polar molecules and the solid particles, and therefore the less ability of the water molecules to move, and that's why their osmotic potential is negative on B. Whereas on A, there are no solid particles at all. There is no attraction force between water molecules and solid particles that might cripple them, and therefore it is considered our 0 reference. So, as a fact, the solute potential of pure water that does not contain any solid particles is higher than the solute potential of any solution. So again, pure water, for example, distilled water, has a higher solute potential or psi S than any other solution, than any other true solution, however small the concentration of the solid particles in it is. Now here we can see a differentiation between, or a comparison and contrasting between, water potential and osmotic potential. Water potential, as we said before, is the measure of the potential energy in the water. I'm sorry, we haven't said this, but why are we putting water potential and osmotic potential in comparison? Because now we are not talking about osmotic potential and solute potential, which are two different ways of expressing the same value psi S, but we are now talking about a comparison between water potential, which is the most inclusive term in that context, or more inclusive, and osmotic or solute potential. So here we have a comparison between water and osmotic potential. You can say here, instead of osmotic, you can say solute potential. And as we said, water potential is more inclusive. So what are we going to compare? We are going to compare the definition, the concept itself, the symbol that denotes it, and the cause. So here it occurs due to, so we are talking about the cause, and you have to be very cautious differentiating between the cause and the effect. So let's read first the content on osmotic potential. It is because we've already covered it. So osmotic potential is just the ability, the possibility of water being able to move from side A to side B, triggered by the presence of solute particles on side B. So osmotic potential is the potential or the ability of water to move from a hypotonic solution to a hypertonic solution. So if I ever tell you that water moved from A to B, then definitely, without reflecting on it, side A is the hypotonic side and side B is the hypertonic side. But of course, we have to add to the definition that there should be what? There should be a selectively permeable membrane. And it is denoted by psi S. And it occurs due to a difference in the concentration of what? Of the dissolved particles. the hypertonic side having more dissolved particles than the hypotonic side. But for water potential, it's a bit different. First of all, it's not denoted by by ipsi-s. It is denoted by, sorry, I apologize again. It's not denoted by psi-s, but it is denoted by psi only. And if osmotic potential occurs due to the presence of the dissolved solutes, water potential occurs due to the movement of free water molecules. And we'll be explaining this. The free water molecules are the water molecules that are not attracted to solid particles. So the osmotic potential refers more to the dissolved solutes, and the water potential refers more to the movement of free water molecules. And it goes without saying that movement applies to free water molecules, because if they weren't free, if they were attached to solid particles, they wouldn't be able to move. You might be asking yourself now, why are we differentiating between dissolved solutes and the movement of free water molecules if the presence of dissolved solutes results or triggers the movement of free water molecules? So it seems illogical to differentiate between them as if they were a difference between water potential and osmotic potential. Actually, it's true that the presence of dissolved solutes results in for example, in the example we have just seen, it is true that the presence of solutes on side B triggered the passage of water or the movement of free water to A. So they should be the same thing, the same cause. Dissolved solutes is a cause, and it results in movement of free water molecules. But why are we differentiating? Because you will understand very soon that the movement of water molecules from side A to side B, and this is very important, is not only triggered by the presence of solute particles on side B, but it is also triggered by other factors like applying pressure on the water molecules of side A. So up till now, we have concluded that there are two forces that might trigger water to pass from A to B. We began by the easier concept, which is the more the solid particles you have on side B, the more the free movement of water from A to B. But studying this comparison taught us that there is another factor, although it's not mentioned in the table, but we'll be studying it in the examples, in the numerical examples. So water potential is related or caused by the movement of the free water molecules, and the movement of the free water molecules can be triggered. Let's erase first. It can be triggered by one of two things, either the dissolved solutes or pressure on the water in the container. And at last, we would come to the definition of the water potential. The water potential is the measure of the energy of the water molecules, or the possible potential. The potential means the possible energy of the water molecules, provided that, in order to make things more accurate, the temperature and pressure are constant, meaning at a constant pressure, at a constant temperature. And this is important to specify the temperature and the pressure so that we would be studying only one factor, which is the possible potential energy of the water molecules. For example, let me give a very small non-numerical example of that concept of energy. Here, for example, if the presence of these 5 solute particles triggers, for example, the passage of 10 water molecules per unit time, say per second, for example, at a certain temperature, which is 37 degrees Celsius, and at a certain pressure P1. So if you increase the pressure on solution A, if you have here, for example, a piston or a flexible membrane, and you push downwards to raise the pressure from P1 to P2, then definitely when you raise the pressure to P2 and you keep the temperature constant and you keep everything else constant, this will help more water molecules to pass. And you will find, for example, that you have 10 molecules of water that pass per second from side A to B. And that's why we cannot say that the water potential equals the osmotic potential only, but it equals the pressure potential in addition to the osmotic potential. And from here comes this famous equation that the water potential, as I said, that is more inclusive, includes the solute potential and the pressure potential. Why? Because it's not only the solute potential that triggers water to pass from the hypotonic side to the hypertonic side, but also the pressure on which side? Mainly on the hypotonic side. If you press on the hypotonic side, as I've just explained, which is side A, then there will be increased flow from A to B. But if you press on the hypertonic side, which is B, then definitely you will diminish the flow from A to B. So again, to wrap up the content of the previous table, the water potential is the potential energy per unit volume of water. This is too broad, of the free water molecules, the ones that are not attached to solid particles. And the water potential is always referred to pure water. And I'll tell you why or how. And as we said before, it should be specified and determined under specific and constant conditions of constant temperature and pressure. It should be specified under constant temperature and pressure. And it quantifies what? It quantifies the tendency of water to move from one area to another. But why isn't it equal to osmotic potential? Because as we have just said, the tendency of water to move from one area to another is not only the effect of the osmotic potential and osmosis, but it is also referred to factors related to pressure, whether it is artificial mechanical pressure that you apply from above or below or the side of the container, and or normal pressure like gravity. And as we said before, it is more inclusive than osmotic potential, because osmotic potential is only related to solute concentration, whereas water potential is also related to pressure. So it is more inclusive because osmotic potential is affected by solute concentration, whereas water potential is affected by both. Is affected by both. By both what? By both the solute potential and the pressure potential, psi S and psi P. Okay? Of course, water potential is very important because it helps us understand how water moves inside plants, how water moves inside the bodies of animals. And you will see this frequently upon studying the physiology of animal and plant cells, or how water moves in the soil. So, solute potential, as we said, is always a negative value, because when we refer it to distilled water, which is our reference, it will be negative. Whereas pressure potential is always a positive value, because whenever you are applying pressure, you are increasing the potential energy of the water molecules to move. So when you, that's why it is positive, when you apply pressure, so you are increasing the potential energy of the water molecules. If they are exactly equal, for example, -5 and +5, then if this is pure water, then the equation is correct because the water potential is 0 of pure water. Of course, you have to specify. So what are the factors affecting the water potential? The main factors affecting water potential are, as we have just said, the solute concentration and the pressure that you apply to the liquid or the pressure of the liquid itself. For a reason or another, every fluid has a certain pressure that is called a hydrostatic pressure. That hydrostatic pressure of the liquid itself depends on many factors. It depends upon the number of fluid particles inside. It depends upon the kinetic energy of those particles and how they hit the walls of the subject. It depends upon the atmospheric pressure itself. But as a general rule, the pressure of the liquid, whether it is an intrinsic hydrostatic pressure resulting from inner factors like the concentration of particles in the container, or from external factors like the atmospheric pressure, it affects the water potential. So let's read together the paragraph. So water potential depends on two main factors. First of all, as we said, let's review the equation. Water potential equals solute potential plus pressure potential. So first of all, it depends upon solute concentration. The addition of solutes to water lowers the water potential. Do we agree with that? Yes, because it makes it more negative in relation to pure water. Why? By convention, and that convention arises from nowhere. No, it arises from the fact that the more solute particles you add, the more attraction force occurs and limits the movement of water. So seawater, or the sap within the cell vacuoles, because it's nothing but water with dissolved salts, will have a negative water potential because they act like solutions, and then pure water. The second thing is the hydrostatic pressure. Hydrostatic pressure, whenever it increases, it will increase the water potential, of course, because the hydrostatic pressure refers to that element of the equation. It makes it more positive, and therefore it increases the tendency. What do we mean by increased water potential? We mean we are increasing the potential energy of the water molecules to move by osmosis across a semi-permeable membrane. And this is how you should say it. You should say it in a very explicit manner. So hydrostatic pressure is the increase in the pressure of the liquid, okay? Or actually the hydrostatic pressure is the pressure of the liquid. It is the pressure of the liquid as a whole, or the pressure of the solution as a whole. It increases water potential because it increases, as we said before, the tendency of water particles to move from the hypotonic side to the hypertonic side across the semi-permeable membrane. So water always moves from higher water potential to lower water potential. Another important concept is what we say here, that we can reach balance in an artificial way, meaning that this is what I have just explained. Say, for example, that if you leave those two solutions without any interference, you would need, for example, 5 seconds to stabilize the equation, or to equalize the concentration of water on side A and B. This is the balance state, or the equilibrium state, the equilibrium state. But if during the process you press on solution B, so you can stop the process earlier, even if the concentration of water molecules is not the same on both sides. Or if you press not on side B, but on side A, you will enhance the water molecules to pass more easily from side A to side B. So you can manipulate the process, stop it earlier, or make it occur more rapidly by changing the pressure on solutions A and solutions B. So if you apply pressure, here I mean apply, not diminish. Let's erase this. If you apply pressure on the hypertonic side, which is here side B, if you apply pressure on the hypertonic side B, so you will stop the flow earlier than expected. You will stop the flow earlier than expected, even if equilibrium hasn't been reached, even if equilibrium has not yet been reached. On the other hand, if you apply pressure on the hypotonic side A, then you are speeding up reaching equilibrium. You are speeding up reaching equilibrium because you are speeding up the process of osmosis. Now, we can see several applications on water potential. For example, here we have pure water. Pure water has a pressure potential of 0 and a solute potential of 0. So the water potential as a whole will be 0 plus 0, meaning 0 millipascal. And millipascal is a unit of pressure. On the other hand, we have in the other arm, we have a 0.1 molar solution and a semi-permeable membrane in between. The 0.1 molar solution has solid particles or solute particles. So its solute potential is negative, which is minus 0.4. But its pressure potential is 0. Actually, the pressure potential on both sides is 0. So here the pressure potential is 0, and there the pressure potential is 0. Why? Because there is no pressure, no external pressure. By convention, if the side of the tube or the mouth of the tube is open and the surface of the solution is just exposed to atmospheric pressure, then we consider the pressure potential being 0. Okay? Now let's see another application on pressure potential, and this application explains the contribution that is made by the application on of an external pressure on pressure potential. Here again we have pure water, and as we all know, the water potential is 0. Here we have particles, dissolved particles that make the solute potential be negative. Say this time it is not 0.4 but 1, which means that we have more solute particles than in the previous example. The pressure potential is supposed to be 0 because the container is open to atmospheric pressure. But actually, why is it positive and not 0? Because it's true that although the test tube is open, yet we are applying external pressure to the surface of the liquid, and that external pressure made the pressure potential be not 0 but +1. So +1 minus 1 equals 0. In this example, we have learned two things. We have learned that it's not because the test tube is open that the solute the pressure potential is 0, because you might apply an external pressure like in this case. The second important concept we have learned is that the pressure potential can cancel or nullify the solute potential if they're equal to each other, because one of them is positive and the other is negative. So they have equal numerical, equal absolute value, but they have opposite charges, one of them being positive and the other being negative. And now we're going to dwell further into the solute potential, because sometimes we don't give you the major components of the equation, which are in this equation, for example, that you see in number 8 on the slide. We don't give you the solute potential and the pressure potential, but instead of giving you a value for the solute potential, we give you the values for the components of the solute potential. So you have to know that the solute potential can be further fragmented into these components: I, C, R, T. I is the ionization constant, and we will say what ionization constant means. C is the molar concentration. R is another constant, which is called the pressure constant, and T is the temperature, because we have said that we have to work at a certain temperature. So what is the ionization constant? The ionization constant is a constant value for every molecule or substance that depends upon the number of ions per molecule. For example, for a substance like the number of ions, or actually more accurately speaking, or more practically speaking, the number of particles in the solution. For example, if we come to a solution of glucose in water, so glucose is non-ionizable, So every glucose molecule will represent one particle in the solution. One glucose molecule will represent one particle, a whole particle in the solution. So the ionization constant of glucose equals 1. When we come to sucrose, for example, sucrose, again, because it is non-ionized, or it is a covalent molecule, the bonds inside are covalent. So despite the fact that sucrose consists of glucose and fructose, but because the bond between them is a covalent bond and the molecule is not split into ions, so one sucrose molecule will also be in the solution considered as one particle, and its ionization constant will again be 1. But in a substance like sodium chloride, for example, you know that in water, the ions are separated from each other because of the presence of the water molecules, and therefore the positive and the positive and negative particles will be separated. So one molecule of sodium chloride, will be separated into 2 particles in a solution, and therefore the ionization constant will be 2. So it is more than, it is related to the number of particles rather than to the number of molecules in the solution. The molar concentration is the number of moles per unit volume. So you will have, for example, a solution of 0.1 molar, and this, of course, is not constant, it is variable. R is the universal gas constant, or the pressure constant. You can also call it the universal gas constant, and it also applies here with the liquids, and it is equal to a fixed value, which is 0.0831 liter bars per mole Kelvin. When we say this means that in the denominator you have moles, you have the the molar concentration, and you have the temperature in degree Kelvin. And as you know, and as you see on the slide, you can change degree Celsius into degree Kelvin by adding 273. So if everything is similar, if you have two solutions A and B, that, for example, a solution of glucose in water, a solution of glucose in water, and a solution of sodium chloride in water. Both are present at the same temperature, and of course the universal gas constant will be the same, and they even have the same molar concentration. For example, both having a 0.1 molar concentration, yet the solute potential and the ability of sodium chloride to attract water will be double that of glucose. This is multiplied by 2. Why? Because the ionization constant of sodium chloride is 2, as we have just explained, and that of glucose is 1. Okay? Here we say, in this window, we say that solute potential is also called the osmotic potential. We explained this before because the solutes affect the direction and the power of osmosis. Psi S, or the solute potential of any solution at atmospheric pressure. We say at atmospheric pressure because in such a case, we are nullifying the effect of pressure that might antagonize the negativity of solute potential, as we have explained before. And we are asking you why it is always negative, because of the attraction forces onto water that diminish the number of free water molecules, and the answer is written on the slide. Now we have worked examples on that slide. In this example, we can see that a 0.15 molar solution of sucrose at atmospheric pressure, and when we say at atmospheric pressure, we mean that psi p equals 0, and at temperature 25 degree Celsius, has an osmotic potential, or a solute potential, of minus 37 bars, and a water potential of minus 37 bars. Actually, this is not a problem. This is a worked example. Let's confirm this. If we apply, here they are talking about osmotic potential and water potential. First thing you notice that they are equal to each other. Let's work out this. We have the water potential equals the solute potential plus the pressure potential. So the water potential equals the solute potential, which is ICRT, plus the pressure potential, which is here a 0 because it is exposed to atmospheric pressure. If you calculate I sucrose, so I is 1 because sucrose is non-ionized. And then you multiply by the 0.15, which is the molar concentration you have, and then you multiply by the constant R, which is 0.831, you will find this value, which is minus 3.7. And minus 3.7 applies to ICRT, which is psi S, or the solute concentration, and also applies to water potential because they are equal values since the pressure equals 0. What is a bar? This unit we are using to denote pressure. What is it? It is a measure of the atmospheric pressure, and it is 1 atmosphere, and it is the same as 1 atmosphere at sea level. So the unit of pressure at sea level is 1 bar. So 1 bar is 1 unit of atmospheric pressure. Now, if you apply the same example with the same temperature, which is 25 degrees Celsius, we forgot here in the equation to add the temperature. Of course, you have to convert it first into, you have to convert it first, 25 plus 273, which makes 198 degree Fahrenheit. So if we apply the same conditions, same temperature, same constant, of course, R, and same molar concentration, 0.15, we will just change the I. And the I here equals 2 because sodium chloride is ionized. So water potential and the solute potential, which are already equal, will both be multiplied by 2. After having studied the osmotic potential and the solute, what concepts have we studied? We first have studied the osmotic. It's important to denote this, osmotic potential. And we've said that osmotic potential is the same as solute potential. And we've also studied the water potential. And then we have studied the pressure potential. So we have studied 4 types of potentials: osmotic potential and solute potential, which actually denote the same thing, and then pressure potential, and then water potential, which is nothing but the sum of osmotic potential and osmotic potential. These nomenclatures are related to some extent to the concept, the new concept we are going to study on this slide, which is osmotic pressure, but they are related to it, but they do not denote exactly the same thing. What is osmotic pressure? We've said before that we have here, if we have here a hypotonic solution A, and there on the other side of the semi-permeable membrane, we have another hypertonic solution B, then definitely solution A has a higher water concentration, solution A is hypotonic, and water will pass from the higher water potential to the lower water potential. But we have said that if we apply pressure here, we can prevent the flow even if the water concentration on both sides is the same. That pressure that you have to apply on the hypertonic solution to stop osmosis is called the osmotic pressure. Of course, the higher the solute concentration you have, the more crippled water molecules you have. And therefore, the more you attract water. So you need more osmotic pressure to stop that water that's coming to you. So osmotic pressure of a solution, after having explicitly explained all the previous concepts, osmotic pressure of a solution is the pressure that needs to be applied to a hypertonic solution to prevent the inward flow of pure water by osmosis. We've defined in the definition a specific type of water, pure water that is able to flow rapidly towards a hypertonic solution. The pressure you will need to apply on that hypertonic side to prevent the flow of not any kind of water, but pure water across a selectively permeable membrane is called the osmotic pressure. And as we said here from this explanation, we can deduce that osmotic pressure is directly proportional. It's not equal to, but the osmotic pressure is directly proportional to the solute concentration. Osmotic pressure has an also more or less direct definition, which is the tendency of a solution to take in pure solvent by osmosis. It seems less evident, but it's used more when you study physiology because it's a shorter definition and it's more easier to remember. Of course, it applies because when you need to apply more pressure on the solution, this means that you have more solute in the solution. And this definitely implies that you're capable of attracting more water. So osmotic pressure can be defined as the ability of a solution to attract water or take in pure water. Osmotic pressure, as we have just explained, is directly proportional to solute concentration because the explanation is written on the slide explicitly and these four statements are actually facts. As the solute concentration increases, more water molecules are attracted. So therefore, we can say that these molecules are crippled and they are prevented from motion. Therefore, this diminishes the water potential in this solution, which is the hypertonic solution, and therefore makes it more liable to receive water molecules from the hypotonic solutions, and this results in absorbing or intaking more water or the more inward flow of water. And in such a case, you would need more pressure, more osmotic pressure to stop the water intake. Having said this, we are very ready now, or we are in a very good position to understand the difference between hydrostatic pressure and osmotic pressure. The hydrostatic pressure is the force of the fluid against a membrane. As we said before, if you have a cell membrane that encloses a cell, this is the best or easiest example we can say. So there is water inside, which is the water of the cytosol. So water is applying force against the cell membrane. That force per unit area of the membrane is nothing but pressure. And because it is pressure, this is the usual formula for pressure in physics, force divided by area. And since this pressure arises from a hydrostatic fluid, since this pressure arises from fluid, then it is hydrostatic pressure. It arises from fluid, then it is hydrostatic pressure. Whereas the osmotic pressure is more related not to the pressure of the container, to the pressure of the liquid on the walls of the container or on the membranes of a cell, but they are more related to the ability to absorb fluid, to take in water by pulling inwards water, as we have said. And this is in blood, for example, it is related to the solute concentration of the blood. So we can talk about the osmotic pressure of blood, which increases with the solute concentration. On the slide, so osmotic pressure increases with the increase in solute concentration in blood, sodium, potassium, and so on. But it also increases when you increase non-ionized particles in the blood, any substance that dissolves in blood or even that is suspended in blood. For example, small protein molecules like albumin and globulin. These are dissolved in the or suspended in the plasma of blood according to their size, and these exert a kind of osmotic pressure, or they define the value of the osmotic pressure of the blood. Why? Because their presence in the blood, although they are big in size, just like the presence of smaller ions like sodium and potassium, these are very useful because they cannot leave the bloodstream, and they help attract water, and therefore they help to preserve the volume of the plasma and therefore preserve blood volume. So they are very important. Imagine that you have fewer albumin molecules in blood, so you will attract less water inside the blood vessel. So the water in the extra cellular fluid here will increase, and this will result in edema or swelling around the blood vessel. So these protein molecules that cannot leave the blood, because unlike sodium and potassium, sodium and potassium exert osmotic pressure that helps inward pulling of water, but sodium and potassium are all the time moving inwards and outwards, whether passively or actively. But such small protein molecules, albumin and globulin, that are actually synthesized by the liver cannot leave the bloodstream, and they play an important role in raising the pressure inside, and they help an important role or play an important role in absorbing or actually not only absorbing but preserving water inside, preserving a certain required amount of water inside the blood vessel, which helps keep the volume of blood and helps minimize the leakage of water outside, which would result in increased water outside the blood vessel in the extracellular fluid and result in edema. But actually, if we want to be very accurate, then we would call this pressure that results from the proteins oncotic pressure. So it has the same definition. It is the pressure needed to stop the inward attraction of water, but it's not generated by small ions or solute particles. It is generated by small protein molecules. If we look at this diagram where we have a blood capillary, and we have blood inside, and the plasma is full of fluid. So you will have an outwards tendency of the water to flow out because of the hydrostatic pressure. Because as we said, the pressure on the walls of the blood vessel, this is hydrostatic pressure. The pressure by the inner fluid on the walls of the blood pressure is the hydrostatic pressure. So the direction of the hydrostatic pressure is always outwards because the fluid inside is pressing on the wall of the vessel outwards. But the pressure, the direction of the osmotic pressure or of the oncotic pressure is inwards. Why is it inwards? Because it results in inwards pulling of water. So you have two antagonizing pressures, the hydrostatic pressure that pushes outwards and the osmotic pressure that pulls inwards. There is a certain balance that is established. And if we have the normal balance between the two pressures, then it's okay. But if the hydrostatic pressure outwards is more, if the hydrostatic pressure, this is H, is more than the osmotic pressure, then we will have an increase in the extracellular fluid and we will have edema. And now we're going to talk about a very simple but interesting example with many applications, which is the tonicity of fluids. But we will stop here and then start talking about tonicity in another video because this video is actually getting too long to be easily uploaded on the internet.